Thank you, Allison. And I have to disclose that I will in no way do justification to the complexities involved in the evaluation and interpretation of these data on behalf of Dr. Goldberger or our key staff member at Tox Strategies, the evidence analyst, Greg Rorby, that was responsible for this outcome. So I apologize for that in advance. So similar to other outcomes, here's the PICO structure. Again, we were evaluating all 4 populations and looking for a variety of, a very broad array of adverse cardiovascular effects across these populations to determine if the non-ROC comparators held true. With respect to the amount of data, this was this is the most robust area in terms of the amount of literature that we found. There were 203 studies included. Of those that were excluded, similar to what you have heard in other outcomes, that was related to the absence of a quantitative metric that we could compare. The majority of data, however, in this outcome were randomized controlled trials. Some were, we had some observational studies, and we had some meta-analyses that we found. Skipping down a little bit to the endpoints, you can see there are quite a few that we evaluated. And in this instance, because we had so many studies, it was important from the front end we took a slightly different approach with this outcome just because there were so many studies, and we classified those readily up front into clinical and physiological endpoints. Now, we still looked at everything. The clinical endpoints were mortality and morbidity. And in many cases, studies would evaluate both. So sometimes it was hard to pull those apart for the purposes of narration. However, those make up our observational studies. So almost all of our observational studies looked at only these clinical endpoints. And along with that, we also had, those were the studies where we were using exposure, dietary exposure from food frequency questionnaires, et cetera. In contrast, the controlled trials used pure caffeine and typically evaluated the physiological endpoints. Listed here, there are quite a few. Another option that we had to pursue in terms of reporting is in the manuscript, we focused on some of the primary endpoints, and the remaining are in the supplemental materials. So starting here with mortality, the highest level, the endpoint ranked highest in terms of decision making, you can see that we had a number of studies, including meta-analyses, and the majority of these support that the comparator is an acceptable intake. This is a situation where we also had some studies that evaluated levels higher than the comparator, though still very few. And it suggested that higher intakes, even up to 855, there were not consistently reported effects. Several studies, and this is for those of you familiar with caffeine research, were at the same time also reporting findings that are suggestive of protective effects. When we looked at the data in totality and looked through our body of evidence, we had a moderate level of confidence in these findings. So now moving into morbidity, as you can see, there were about the same number of studies as we had for mortality. And in this instance, we all had a number of studies that were reporting on morbidity associated with certain genotypes and phenotypes. So this is an example where we might have multiple studies reported on a given graph. When we looked at across these studies, our ultimate conclusion was that 400 mg per day was an acceptable intake that was not associated with significant concern, but as similar to what Dr. Peck noted, is that there were some observations where individuals with certain sensitivities may be more sensitive to some of the effects. In this is also important an important figure because we had a fair amount of studies reporting above the comparator, and as you can see, those were associated with a lack of effects. So these inconsistencies, again, make it difficult to integrate these data when you're finding a lack of effects above the comparator and a report of effects below the comparator. There was a low level of confidence, or moderate level of confidence in this evidence-based, primarily influenced by the low level of indirectness in that they were, i.e. that's a double negative, but they were direct, they were good measures of our studies, and an overall low risk of bias. Blood pressure. I wanted to make sure that we showed this one because this, I think, highlights some of the complexities involved with this review and interpreting and integrating this data in a more of a risk-based context. So, similar to other endpoints that you've seen, the majority of data that we have to look at were below the comparator. And this is only a snapshot of the data. We had quite a bit of blood pressure data with the 3 plots in the manuscript showing this. And again, the majority of these are from controlled trials, which were evaluating oftentimes performance events. And this took a lot of discussions, many discussions with Dr. Goldberger to interpret these data. This is also an example of when I mentioned earlier that we knew that we were going to be faced with a fair amount of data that we needed to be able to talk about if they were adverse. And in many of these cases, the idea of adversity is contextual. But what we found for a number of the points of comparison below the comparator where you're seeing orange is that we're only seeing a few milligrams of mercury change in blood pressure. So they're very small. In fact, these are smaller than the amount that you're seeing just daily in your variation in your blood pressure throughout the day. So the magnitude of change was small, and the biological meaning of those seemed to be quite small as well. In addition, these were transient changes, and the transient increases sometimes can even be helpful. So, for example, when you're exercising and your blood pressure increases, those are actually regarded as a beneficial effect. So that added complexity to interpreting these data. Additionally, these short-term transient changes on blood pressure, it's unknown what their long-term effect is on, for example, cardiovascular disease. So at the end of the day, this forced us to think quite a bit about what these data show, and we ultimately concluded that this comparator, clearly you can't argue that it may be too high if you're only considering the potential for caffeine to cause a physiological change. But when you're thinking about the more complex picture in terms of changing biology or potentially adverse effects, it was our expert judgment that this comparator was acceptable, and this was based primarily on the small magnitude of changes and this transient nature and the lack of knowledge if these transient changes have any downstream impact on cardiovascular disease. Because our evidence base for cardiovascular was primarily controlled trials, again, mostly looking at the physiological endpoints, so blood pressure, heart rate variability, the overall confidence in the evidence was high, given that it had a probably low risk of bias, or moderate to high, excuse me. The primary limitation that we saw was in the observational studies and the exposure characterization. So similar to other outcomes, when you had the observational studies and you were thinking about how the exposure was characterized, again, the dietary surveys and having confidence in how much was reported was our main area of uncertainty. So shown here, we've got the confidence table, and as you look down the endpoints that were evaluated for both adults and adolescents and children, I didn't put the figure up, but we did have some data characterizing such in those populations. You can see that we had a moderate to high level of confidence, initial level of confidence, and that the majority of these studies were developed or designed to evaluate our questions. So they were giving us very direct information on our outcomes. So that increased our level of confidence. Similarly, there was a low risk of bias across these studies, which also increased our confidence. With respect to confounding in primarily our observational studies, this is where it came into play the most. There was a relatively high degree of consideration for confounding variables. With respect to blood pressure and a lot of the RCTs, there were not a lot of considerations per se, as you might expect, for confounding, but there was consideration for some of the concepts that we've just heard Dr. Lieberman and Dr. O'Brien talk about with withdrawal and how if you're coming into it naive, or the considerations in the study design to accommodate for those factors. And in the end, we had confidence ranging from moderate to high in these study types. And then, again, not to make you squint and look at the table, but just a reminder, all the information in the table, that's essentially showing our work as to how we develop these conclusions, demonstrating where we have data, where on the spectrum we have data, and where our conclusions lie. So based on the weight of the evidence, when all factors were considered, particularly our emphasis on our level of adversity, so again, using blood pressure as a comparison to cardiovascular morbidity and mortality, for the 400 mg per day was found to be an acceptable intake, not associated with significant concern on cardiovascular effects. Clearly, we did see that there were findings suggesting the comparator was too low. Those were some of those plots I showed at the beginning, where you had exposures up to almost 900 with a lack of effects. And then we also had findings below the comparator, and these were mostly the physiological endpoints: heart rate variability, heart rate, blood pressure, which you were seeing impacts below the comparator. With respect to children and adolescents, it was ultimately determined that the evidence base was insufficient to develop a robust conclusion, and that the available data we saw were somewhat inconsistent, which also led us to hesitations in developing conclusions for these populations. With respect to future research, as with many of the other outcomes, we'd like to see more research in children and adolescent populations. One of the topics that came up quite a bit was the temporal exposure response, so gaining an understanding of the dose-response relationships following chronic exposure for some of these endpoints would be helpful to know if these transient effects are meaningful, et cetera. And then something that was a theme certainly throughout the assessment is a better characterization if what or any magnitude of these changes would be associated with adverse effects, something that is probably a research project on its own for this topic.